you, Denver and Brent. It's a pleasure to be here, and only having to travel 15 minutes to get here makes it a delight to go to conferences. So, if you want me to go to your conference, have it in San Diego. So, aside from a shameless plug about my book, this is also the title of my lecture, and I'll take you through the path that we've gone through over the last about a decade. First, digitizing the genetic code. So when we sequence DNA, we convert the ACs, Gs, and Ts into the ones and zeros of the computer world. And I refer to that as digitizing biology. And that started for us in earnest in 1995 when we sequenced the first genome of a living cell, and what more of you are familiar with the first draft of the human genome in 2001. But with the change in the scale of sequencing, my genome cost about $100 million to sequence. Now Illumina can sequence a genome for a little over $1,000, so things have changed quite dramatically, and the amount of data changing in the databases has changed relevant to the cost of that. So we started to look about going the other way. Could we start with the ones and zeros of digital biology, use that to redesign and make the chemical code, and then find a way to boot that code up. And so this started with asking simple questions after we had the first two genomes of how many genes are essential for life, and could we get to a minimal cell number, a minimal gene number to aid in future genome design as we move into the design phase of biology. And to answer these questions, we ultimately wanted to see if we could design and construct a minimal genome of a living cell. But nobody knew whether the chemistry would allow this to happen, and we had no idea, even if the chemistry worked, could we find a way to boot up the chromosome. So we set up two different teams to work on this. My leading colleagues in this are Ham Smith, who got the Nobel Prize in 1978 for discovering restriction enzymes. He's 82 and works every day in the lab. And Clyde Hutchinson, who retired from UNC, is 75 and also works every day in the lab. So if you want really good postdocs, hire retired professors. They actually know what they're doing and they work hard. So, but we started on this problem, and we decided to start with something small. We chose Phi-X 174 for several reasons, including its historical significance as the first DNA virus that Sanger's team had sequenced. And Clyde Hutchinson was a member of Sanger's team and took part in that sequencing. But it was also known that Phi-X can't take many mutations and still get active virus out. So many of you know that DNA synthesis is an error-prone process. With standard synthesis, it's an n-1 situation, so the longer you make the sequence, the more errors you have. So we developed a number of new approaches for error correction. We started with the 5,300 base pair sequence. We made small pieces, put them together, corrected the errors, and then injected that piece of DNA into E. coli. And this was the actual first experiment with this. Basically, E. coli took this DNA up, started reading it immediately, started making the viral proteins. The viral proteins self-assembled, formed the virus. Once a certain number of copies are made, it kills the cell, and that's how we detected these clear spots, or where a large amount of bacterial killing had been done by a virus that had been made. So we call this a situation where the software builds its own hardware. We just put in a piece of DNA software, and we got out these viral particles that are capable of infecting E. coli cells and making lots of copies of themselves. So we knew at least we could master some of the basic principles, but whether they would work on a larger scale was unclear. But we figured if we could make viral-sized pieces of DNA accurately, we could put hundreds of these together and try to stitch them together to make a bacterial chromosome. And so the team worked on this in a pretty slow process. We made these viral-size genome cassettes of 5 to 7 kb. There was 101 of them. Something that looks like a Final Four playoff. We put 4 of these together, to get 24 kb pieces. And after we did this, we had to clone them in E. coli, grow up a lot of it, sequence it, make sure we had not introduced new errors, because nobody had been down this road before, so we wanted to make sure processes we're doing wouldn't add errors or complications to the genomes and to design. And after we did that with all the different pieces, we then went on to the next stage of putting 3 of those together to get 72 kb pieces. And at this stage, we exceeded the world record by a substantial amount. Over a many-year effort, one team put a 30 kb gene cluster together. It was a heroic effort, but was not something that was really replicable in genomics. After the 72 kb level, again cloning these, sequencing them, we went on to make quarter molecules, 144,000 base pairs. And at this stage, these big pieces of synthetic DNA weren't so happily cloned into E. coli. So we started looking around for a new system, and we chose Saccharomyces cerevisiae. We found it could take up very large pieces of DNA, maintain them stably, but even more importantly, you can put them together readily by homologous recombination. So that's what we did. We put these 4 quarter molecules, quarter genome molecules into yeast, along with just a small vector that contains a synthetic yeast centromere, allowing replication of this new chromosome along with the rest of the yeast chromosomes. Homologous recombination put these all together, and we ended up with the first assembled synthetic bacterial chromosome. And at the time we reported this in Science, this was the largest chemical of a defined structure ever put together. The team continued to work on the synthesis methods. And Dave Gibson, who started out as a postdoc in the lab, took what we were doing in a very laborious set of methods and combined them all into a single tube at 50 degrees centigrade. And because this works so well and has changed what we and everybody else can do, that we now call this the Gibson assembly. And basically you can just put oligos or small pieces of DNA into this tube with the 3 enzymes, incubate it for a while, and you get nicely assembled DNA, which means this process could be further automated. And so we put a lot of effort into doing that, going from the digital world of the computer into making DNA pieces in an accurate fashion. So while synthesis was proceeding extremely well, we had a second team that had been working on what proved to be a more difficult problem. How do you boot up a bacterial chromosome? And so we had to develop a technique of genome transplantation. In this paper, I referred to, there's a whole chapter in my book about this, was I think one of the most important experiments, set of experiments that I've been involved with that really explained to me how biology works. And because this is such an important paper to what we do, I thought I'd walk you through it a little bit. We isolated the genome from one species of mycoplasma, M. mycoides. And when we first did this, we had no idea whether there were going to be proteins required for transplantation. Could we actually use naked DNA? So we treated the genome harshly with proteinases to remove all the proteins. We added a couple of cassettes so we could select for the chromosome, and it would turn cells bright blue if it got activated. And we had to work out new techniques for moving genomes around. Large pieces of DNA like this, you can't pipette them. The shearing forces of pipetting will just shear the DNA, and large pieces of DNA are actually quite brittle. So we worked out methods to isolate the chromosome in gel blocks, and you can move enzymes in and out of the gel blocks. And then for transplantation, we actually use an enzyme to dissolve the gel and use electroporation, amongst other techniques, to get it into the cell. So to show you what we think happened, we have this really sophisticated movie. So we inserted the mycoides chromosome into the recipient cell. The recipient cell is M. capricolum, which is about 11% different, a little bit more different than we are from mice, and a different size size genome. So at this stage now, we have the embodiment of one species, so everything you would measure about species you would find physically in that cell, but it now has two sets of genetic software. What we think happened as soon as we put in the new chromosome, as with the Phi-X genome, it started to be read immediately in producing proteins. And some of the first proteins produced were restriction enzymes that recognized the capricolum genome as foreign DNA and chewed it up. So now we have the body of one species and the genetic software of another. So what happened? In a very short period of time, we ended up with these bright blue cells. And then when we interrogated them, there wasn't a single molecule left of the original capricolum species. Every protein was derived from the mycoides chromosome that we put into the cell. All the antigens switched over. There wasn't anything detectable of that original species, which leads us to the notion that life is a DNA software system. And if you change the software, you change the species. This actually has a lot of implications for evolution, because when we were sequencing microbial genomes around the oceans and around the world, we found multiple species that had more than one chromosome. We discovered this first actually with the cholera genome. When DOE was funding us right after our first genome to do a number of genomes, we wanted to do cholera, but the late Carl Woese argued that the cholera genome had basically the same 16S RNA as E. coli, and we wouldn't find any differences from E. coli. With the shotgun sequencing, you don't have any biases, and it assembled the DNA into two separate chromosomes, one which was very similar to E. coli, and one which was very different and didn't look anything like E. coli. So what can happen when species have mismatches in their restriction systems and fuse together? You can add thousands of traits to a cell in a single evolutionary event. So instead of evolution just being a series of quaint mutations and selection, it was also a series of major events of merging entire genomes to add, as I said, thousands of traits. In fact, that's how the plant world developed, by a small cyanobacteria getting absorbed into a eukaryotic cell and becoming the photosynthetic center. And also for mitochondria was a bacteria that got absorbed in to a eukaryotic cell, and both the chloroplast and the mitochondrial genomes are still maintained separately in cells. So we see evidence for this type of major evolutionary change. Now, we had a problem that we were assembling the bacterial chromosome in a eukaryotic cell in Saccharomyces. So we had to find a way to isolate the chromosome from yeast to be able to transplant it back into the bacteria. So to work out these tools, the team developed ways to clone entire bacterial genomes as yeast artificial chromosomes. In contrast to what the literature said early on for the attempts to do this with human chromosomes, these chromosomes are totally maintained in a very stable fashion and just get carried along as an additional chromosome in yeast. And we've done this now with a wide range of microbial genomes. So we had the ability now to test isolating the normal mycoides genome from yeast and trying to do a transplant. The trouble is, it didn't work. And it took us almost 2 years to fully solve this problem. That in retrospect should have been a lot easier, seeing that we had the Nobel laureate who discovered restriction enzymes in the first place. It turns out in the mycoides cell, the genome was methylated to protect it from restriction enzymes, its own and a few others. So when we purified from cloned DNA the 6 methylases and methylated the genome after we isolated it from yeast, all of a sudden the transplants worked again. And to ultimately prove this, we removed the restriction enzymes from the recipient cell, and in that case we could just take naked DNA out of yeast and transplant it. So we put all this together in something that gives us unique tools